Okay, so today we're going to discuss a bit on the questions, which is a describe of cyclic photophosphorylation in hibiscus and explain the carbon dioxide fixation in CM plant. Okay, before we go further in the discussion, bear in mind hibiscus is the scientific name. So how to write scientific name? Hibiscus has to be underlined and SP dot no need to underline. Okay, so this is the writing of scientific name. Okay, so before we discuss in detail how to answer this question, so bear in mind cyclic photophosphorylation. When we're talking about these questions, you should actually come out with the diagram which is suitable to this particular answers. Okay, also recall the carbon fixation, the CM cycle diagram, and the Kelvin cycle diagram. So there will be three diagrams that we're going to use today: the cyclic photophosphorylation diagram, Kelvin cycle diagram, and also the CAM cycle diagram. Okay, so this is the diagram that we use for the cyclic photophosphorylation. So we have the involvement of photosystem 1, the involvement of primary acceptor the involvement of the primary acceptors. Okay, so the primary acceptor, then the ferredoxin here, and then we have the cytochrome complex, the plastocyanin, and then we have the ATP production by chemiosmosis. Okay, so at the photosystem here, the process involved is photoactivation. Okay, photoactivation. So by referring to all the information here, how to plan our answers. So first of all, this is actually happen at where? This is actually happen at thylakoid membrane. Okay, so the first point is come from the thylakoid membrane. State the location first. It is happen at thylakoid membrane. Cyclic photophosphorylation. What is the aim of the cyclic photophosphorylation? Is to produce more ATP. So to produce more ATP. To support what? To support the process of the Kelvin cycle later on. So another 1 point. So it's already 2 marks. So the explanation at the bottom here will only have another 4 points. So other than location and aim, the discussion of process is only have 4 points. Just now we already mentioned photoactivation, and then the entering of the electron transport chain, produce ATP by chemiosmosis. So almost have 4 marks already. So how to write the essay question? So it's no need to be a very detailed essay question. Okay, so we just go through in a very fast forward what is actually need to write. Okay, so first of all, we actually need to mention what happened at photosystem 1. Okay, so photosystem 1, will what will be the main purpose of photosystem 1? So photosystem 1 absorb light energy. So this is the first step. Okay, photoactivation, photosystem 1 absorb light energy. Okay, so this will so the energy absorbed, what will happen to the energy absorbed? So the energy absorbed will photoactivate 1 electron from the primary electron acceptor, from the chlorophyll A here into the primary electron acceptor. So how to write? Okay, so we have to mention so the electrons. Okay, so the electrons, so the energy absorbed will pass to the energy absorbed will pass to the reaction center. So who are the reaction center? The reaction center, the P700. So this is another 1 marks. Okay, then at the reaction center, the electrons. So the electrons in chlorophyll A, so absorb the energy and get photo excited. Okay, when they get photo excited, what happened? So when the electron get photo excited, it enter the higher electron energy stage. So the electron accept by the primary acceptor. Okay, so enter higher energy stage. Then the electron received by the primary acceptor. Another 1 marks. So the primary acceptor pass the electrons into the electron transport chain, 1 marks. Electron transport chain is built out by ferredoxin. Okay, so a series of redox reaction is take place from the ferredoxin to the cytochrome complex, then pass to the plastocyanin. Then the electrons will move back into the reaction center of photosystem 1. So this is the component. So the component, you mentioned ferredoxin, cytochrome complex, plastocyanin, and finally end up at photosystem 1, you get 1 marks. Okay, so the electrons, so then ATP will produce through the chemiosmosis that take place at the thylakoid space. Okay, so then that's it for the process of the cyclic photophosphorylation. Then we go into the second question, which is about the CAM plant. Okay, so when we're talking about the CAM plant have 2 pathways. One is the CAM cycle, another one is the Calvin cycle. Both undergo carbon fixation. So both undergo carbon fixation. So how to explain about the carbon fixation and where does carbon fixation take place? Both happen at mesophyll cell. Okay, one happen at night, one happen at day. Okay, so this is the general information we can generate. So how to write the carbon fixation? Okay, so bear in mind, question B is only want us to discuss about carbon fixation. So no need to discuss other than that. Okay, we only focus in carbon fixation. So it happened. Okay, so carbon fixation of the CAM plant happened at night and happen at day. So we just mentioned what happened during night, what happened during day. So at night, stomata open. Okay, so this one, 1 marks. So carbon dioxide open, carbon dioxide enter the cell. So this one, we're going to give you 1 marks. Okay, so when carbon dioxide enter the mark, it will bind with the PEP, 1 marks. Okay, forming oxalo acetate, 1 marks. So this happened at the mesophyll cell, 1 marks. Okay, and this process is helped by PEP carboxylase, 1 marks. Okay, so then, okay, so this is the part of the carbon fixation during night. So then we go to the carbon fixation during day. So at day, stomata is closed. Okay, stomata is closed. So no carbon dioxide from surrounding enter cell. Okay, so this one is 1 marks. Then it will cause the carbon dioxide concentration reduce, 1 marks. Okay, so then we discuss lah. So this will trigger malate to release carbon dioxide. Okay, this will trigger malate to release carbon dioxide. Then what happened next? So once malate release carbon dioxide, so the carbon dioxide will fix into the, will bind with, so the carbon dioxide will bind with the RUBP, 1 marks, to form the temporary 6 carbon compound, which will immediately split into 2 molecules of 3-phosphoglyceride. Okay, forming into temporary 6 carbon compound, which is immediately split into 2-phosphoglyceride is also 1 mark. This process is catalyzed by Rubisco, 1 marks. Okay, so 1, 2, 3, 4, 4 marks here, adding to the 4 marks, another 4 marks here, 1, 2, 3, 4, 5. So enough for the 9 marks question. So this is how you answering the question relate to the question A and also question B. Okay, do we need to actually discuss the whole thing on the CAM cycle? The answer is no need. But if you don't know how to answer this question, then you can explain everything on the CAM cycle, and then also don't forget the pathway on the Kelvin cycle, the carbon fixations that happen in the Kelvin cycle. So that is for answering this question. If you still have any question, ask inside the group. Okay, so that's it for today's discussion.